Dear all, welcome to Civil Services with ZAKI, and today we are going to discuss about the RNA editing and its significance. As you know, it is a recent phenomenon and very significant regarding the gene editing. And as you know, in the recent year in United States, Food and Drug Administration basically approve the clinical trial of RNA editing. Basically, these RNA editing are related to CRISPR-Cas13. So, what is CRISPR-Cas13? What is RNA editing? And how these type of RNA editing work? And also, we know the basics of the RNA, DNA, and what is the protein synthesis and all that. So, basically, these video all our previous video, as you see here, like the Indian Genome Project and all that, we also provide you the several video on the science. So these are very significant video regarding the prelims and as well as mains. So in the last we also discuss about the mains question regarding the RNA editing and also PT questions. So that's why please stay with us till the end of this video. And as you know, first we start with the subtopic. So today's subtopic are number one, what is gene editing? First we understand the what is gene editing. First, what is gene editing? Another is what is RNA? What is gene editing? What is RNA? First, before we going to start the what is RNA editing, first we understand what is RNA, and another is why is the synthesis of protein is important. Basically, protein synthesis is the one of the significant important process in these editing. So, what is protein and why these proteins are very significant? We also discuss that point. And another is what is RNA editing. Then we move towards what is RNA editing, and finally we see what are the ways to edit RNA. So what are the ways to edit RNA, what is RNA editing and all that. Another is if we see what are the editing application. One of the most significant applications is called repair and rescue. So what is repair, what is rescue, and basically these are genetic material editing tools. So it is very significant. Basically, if you see all these points are in the beginning state, but in future it will be going to the very significant in the basically the field of medical, genetic engineering, all that. So that's why we you stay with us. Another is what is the significance of RNA editing. So why RNA editing is significant than DNA. So what is the difference between the DNA and RNA editing? And another point is what are the challenges associated with RNA. So some of the challenges, like some of the recent digital event is still awaiting for the future research and all that. So as you know, RNA editing is in its early phase, so that's why we also take time. So some these type of the points. So if you want to know all these points, then stay with us. So first we see what is the context. So regarding the context, we see the United States Food and Drug Administration. If you see here, Food and Drug Administration has just approved the first ever clinical trial. So basically, why this topic is new? Because it is first ever clinical trial uses CRISPR-Cas13 RNA editing. So it is used, as you know, the CRISPR is the one of the most gene editing tool. So it is CRISPR-Cas13. So basically, it is related to the wet age-related macular degeneration. In short, you can call it AMD. So these AMD disease are basically related to the eye, so causes vision loss in the millions of older people across the globe. So recently CRISPR technology has, if you see the CRISPR technology revolutionized the field of genetics, allowing for the precise, so it is precise editing of DNA or RNA. So while most discuss, if you go to the go to the across the website and all that, you can find easily CRISPR and DNA technology. So CRISPR-Cas9. So as you know, it is very renowned genetic editing tool. But edit DNA. But CRISPR-Cas13 is specifically designed for RNA editing. So that's why RNA editing is one of the most significant tool in the recent development. So let's see first what is gene editing. So first we understand what is gene editing. So if you see here, gene editing is also known as genome editing. Okay, so it is set of the technology that enables scientists to make precise alteration of the DNA of living organism. Basically, if you alter the DNA, basically the base pairs of the DNA, so these processes involve basically, if you see, the adding, removing or modifying. So adding, removing and modifying the specific sequences of the DNA and targeted location within the genome. So you can target such of the bad base pair and replace it with the your desired base pair. So it's also used for the genetic engineering, disease treatment and all that. But if you see, the gene editing tool basically regarding the DNA is a permanent change. So to the person's DNA. So DNA editing is permanent. So if you see the DNA editing, so if you altered some person's gene, it means maybe it's permanent. But in RNA event, on the other hand, it is transient. It means it's temporary because RNA molecules are consequently being made and degraded in our cells. So if you see our cells, RNA molecules continues made and disintegrates in our cytoplasm or cell. So let's we see what is RNA. So why RNA is important? First we know what is RNA. Basically RNA is ribonucleic acid. If you see the RNA, it is the short form of ribonucleic acid, RNA. So it is ribonucleic acid. It is called RNA. Basically, it is a single-stranded biomolecule. It's play a very significant role in the various biological process in our cell, particularly in the synthesis of protein. So basically, RNA is very significant for the synthesis of the protein and regulation of the gene expression. Two things very important played by the RNA. So why synthesis of protein is very significant? So we also see here RNA. So see, this is DNA, this is RNA. This is DNA, this is RNA. If you see the RNA and DNA share same three base pair, like the cytosine, guanine and adenine. Cytosine, guanine and adenine. Cytosine, guanine and adenine. But the difference between the RNA contain uracil and DNA contain thymine. RNA contain uracil, DNA contain thymine. So that's the difference. So basically, if you see here, this is RNA single stranded and DNA double stranded. So I hope you understand the RNA. So why protein synthesis is important? Because if you see protein synthesis, fundamental biological process. So if you see the synthesis of protein is the fundamental biological process, play crucial role in the functioning and maintenance of our living organism. So it's also functioning and maintenance. So some type of your cell degenerate and degrade. So maintenance, work of the maintenance is also done by these protein. And also if you see, the protein are composed of long chain of amino acids. Basically if you see the regarding the human, about 20 amino acids. So which fold into the specific three-dimensional structure and determine their function. So it's basically protein composed of the amino acids, and these amino acids shapes a specific 3D structure. So if you see the protein are integral parts of the immune system. So this is the very significant for our immune system. It protect against pathogen. So protein synthesis is also direct related to the gene expression, so allowing cell to respond dynamically environmental changes or development cues. So it's also directly related to the gene and also expressed with the environment. So that's why protein synthesis is very significant thing. So let's we move forward and see the formation of protein in an animation. So see here animation. So if you see here, basically if you see here protein synthesis. So as we know, the fundamental unit of every living being is the cell. If you see cell is our fundamental units. Another if you see here cell contain various organelles, various parts like the mitochondria, Golgi body and etc, but it has a nucleus. And so if you see here nucleus, then in nucleus contain your genome and nucleic acid. So each and every genome contain chromosomes, and these chromosomes like our humans and all that, about 23 pair of the chromosomes, and these chromosomes if you see here, its chromosome contain DNA. Basically these DNA tags are bound with the protein called histone. See here, it is histone and it is DNA. If you see here DNA, but if you see each DNA contain gene, it's called gene. These base pair if you see here, it's called gene. These base pair, so these base pair are known as gene. So basically the modification of these genes like the base pairs is called genetic engineering and all that. So but it play a very significant role for the formation of the protein. See here, these DNA go to the an enzyme called RNA polymerase, and these RNA polymerase basically translate into the messenger RNA. So see here, it's here, and it is base pair. All these base pair like the adenine, guanine, cytosine, all base pair. See here, all base pairs. So this is known as messenger RNA. So if you see these messenger RNA, naturally basically in edited and response to the environment, and finally after these editing, these messenger RNA go to the cytoplasm, and then finally from cytoplasm it's go to the protein factory. Protein factory is ribosome. So it's also known as, if you see the what is known as the protein factory. So ribosome known as the protein factory. So go to the ribosome. So these messenger RNA goes to the ribosome, and finally produce amino acids, and finally these amino acids formed by these type of the RNA. Basically it's called transfer RNA, and basically it has 3 base pair in this area, and finally these amino acids finally make all these amino acids, and finally when completed 20 amino acids, it's finally go to a 3D type of this and make protein. So that's why this protein is basically these protein. So it's protein. So these proteins are play a very vital role in the life process in the human being and all organism. So let's start with the what is RNA editing. Is our topic. So what is RNA editing? So RNA editing is a process of modifies genetic information of RNA molecules. Regarding that, so it means changing the sequence of RNA molecules by adding, removing, or substituting nucleotides. So these nucleotides, as we saw, all genomes, strands of the RNA and DNA, all including known as the nucleotides. So nucleotide it can occur in the nucleus, mitochondria, plastids, and many organisms. So if you see, it's on mitochondria, plastids, and many organisms, including the human, plants, viruses, etc. So it can present human, viruses, and all that. So some of the, if you see, some of the viruses contain RNA as a genetic material, like COVID virus, most famous in recent year. So let's see what are the ways to edit RNA. So what are the different ways to edit RNA? So as you know, this is RNA editing is in its early phase. So in early phase, but some of the basics editing tools present in the world today. So if you see, RNA editing technology has gained traction as a promising tool for the therapeutic application. So basically it is particularly in the gene therapy. So basically it is used as a gene therapy because it is not damage or create some side effect permanently, because it is temporarily. So there are four primary ways to edit RNA. So if you see these four, what is the, so these are, what is four? First, if we see, it's called base substituting editing. So it means you can substitute the base. So type of involve the changing of nucleotide base in another RNA molecules. So basically it is the most common editing tool is adenosine inosine A to I editing. So it is most common A to I editing. So where if you see adenosine converted into inosine, if you see the inosine interpreted by the guanosines Z during the translation. So basically the messenger RNA translate guanine as inosine. So basically potential altering the resulting protein amino sequence. So basically it is change the sequence of the amino acids. Amino acids. So this process primarily mediated by the enzyme. So basically what is the enzyme? It is called adenosine deaminases acting on RNA. In short, the enzyme name is called ADRS. ADRS enzyme basically found in human body. So see here. So what is mechanism of this editing? So if we see ADR recognize double stranded RNA. Basically if you see the RNA, so RNA is a single stranded, but it's not so straight. So it means they can curve this type and make this type of the double stranded RNA sometimes. So it can change significant protein functions, changes in the, so after that it changes the code of the genetic proteins functions. So it is a change of the codon. So first we see the animation. So how this technique, basically ADR's techniques work. See here. in animation. How does ADR work? So if you see here, ADR is a very powerful tools to basically treating diseases, such type of the diseases. If you see the such type of the brain diseases, liver diseases, heart diseases, even lungs diseases. So all these things are treated by this ADR technique. So it is one of the most significant technique. So see here. So first, as we know, the DNA transcription into the RNA, and finally RNA translated these into the amino acids and proteins. So see here, DNA, RNA and protein. This is protein. So DNA transcription to the RNA, and RNA finally translation to form protein. So basically these type of the gene editing, it's called A to I gene editing mostly, and basically it's form translated into protein. So with this type of the protein and basically desired protein, so basically it is the A to I editing, adenosine changed into the ionosine. See here. So how does this work? So if you see here, this type of this is RNA. If you see here, this is RNA molecule. So this is double-stranded RNA, and finally if you see here, these ADR enzyme finally use and fix the A to I to convert A to I. Further, it's another ADR used. Finally, it's edited. So basically, these ionosine, basically it's guanosines. So A equal to I equal to G. So finally, they prepared the functional protein, and finally these functional protein finally accomplish A to I editing, and if you see here, it's work like a virus helping immune system. As you know, protein is directly related to our immune system, so it's work like if you use laptop and another electronic device, antivirus. So it's look like antivirus mechanism of the protection. So help immune system to protect our body from the viruses and the bacteria. See here, from viruses and bacteria. If you see the normal human genome and another normal human RNA and virus human RNA. So finally, if you see in this technique, it's virus and it's virus RNA and it's human normal RNA. So these technique, these ADR identify these viruses and finally kill this virus or such type of the pathogens, virus or bacteria, and finally remove from our cell. But as you know, it's fixed adenosine to ionosine, and finally these basically enzyme basically not activated in the normal RNA. So basically these technique used in the various basically vital role in the neuron. So as you know, the neurons is basically very significant in our brain. So basically neuron and all these things, basically these are the two examples, but also if you see the some RNA, see here. But one of the major things, if you see here, This is ADR. ADR is very significant to tools to prevent our immune system or other protection from the diseases, because if you see in our body, various type of the ADR present, and this is very significant for the protect our various organs, so multiple disease area. So it's a great potential for the future. Another is most significant, second is CRISPR-Cas based approaches. This approach is also one of the most significant approach. So CRISPR, basically what is CRISPR? CRISPR is, if you see, clustered, regularly interspaced short palindromic repeats. Palindromic repeat. So cluster, regularly interspaced short palindromic repeats. In short, it's called as CRISPR. So CRISPR-Cas13 system is one of the. What is Cas? Cas means CRISPR associated protein 13. So if you see Cas9, it means CRISPR associated protein 9. What is Cas13? CRISPR associated protein number 13. So protein 13 is specific for the RNA editing. It is a revolutionary tool. So offering a new opportunity of the development of the nucleic acid therapeutic. So it is also very significant because the first ever clinical trial of the RNA, means AMD, age-related macular degeneration disease, basically a trial for this disease. So if we see here mechanism is very significant. If you see, basically it's consists a single multifunctional Cas13 protein and a guide RNA. Guide RNA, it's also called CRRNA. So what is this CRRNA? CRRNA means CRISPR RNA. And these CRISPR RNA complementary to the target RNA and sequence, allowing the Cas13 bind and cleave a specific RNA target. So it can bind and it can cleave a specific target, RNA targets. So if we see another thing related to these CRISPR-Cas13 is also called RNA editing application repair and rescue. So what is repair and rescue? So if we see in the CRISPR-Cas13 system adapted for the RNA editing applications such as repair. Repair means RNA editing for the programmable A to I replacement. So it's also called, if you see the A to R replacement, it's called repair, and from a specific C to U exchange, it's called rescue. Two things is very. So we also have some animation about the CRISPR-Cas13. So see here animation of the CRISPR-Cas13 technique. So what is this technique? See here. CRISPR-Cas, as you know, the CRISPR-Cas9 is specific for the editing of the DNA editing. So what is CRISPR? CRISPR is clustered, regularly interspaced short palindromic repeats. In shorts, it's called CRISPR. Cas19 protein. So if you see here CRISPR-Cas19 protein here, it's Cas9. So if you see here, it's need two. Number one, Cas19 and guide RNA. So one of the guide RNA, another is Cas19. So it's very significant to edit the DNA base pair. If you see here, like these base. and find target and finally replace the genetic base pair with the other base like A to G. So basically, see, this is so this type of the editing. Basically, these type of the editing practices in the DNA editing, and as you know, the DNA editing is the one of the most significant in these times. But if we see here RNA editing, we see RNA. So it's a Cas9, basically for DNA. So cousins of Cas9 is Cas13. Cas13 is the another protein enzyme for RNA, basically editing for the RNA. So see here, it's called. See, when these Cas13 activated, it's work like a Caesar, like you see, Caesar. And this type of the process called collateral cleavage. Basically, this type of the process happens in the nature, but also scientists use this type of the technique to treat diseases. So see here. So this type of related diseases used this type of the technique used in the recent clinical trial related to basically wet age macular degeneration diseases like the eye diseases. So this is very significant achievement in the history of medicine or DNA editing or genetic editing. So see here. So this is all this. So another third is cytidine and uretidine C to U editing. It's also another technique to basically edit RNA, but these technique or also play a very significant role, basically cancer and autoimmune disorder. It play a very significant cancer and autoimmune disease. Mechanism basically mediated by the protein called apobic protein. Apobic protein which target special RNA sequence. Another fourth is also significant insertion or deletion or editing. So it means you can insert base pair or delete base pair. It's also known as indel. So indel editing is a specific type of RNA editing involved in addition or removal of nucleotide with the RNA molecules, basically resulting changes in the encoded protein sequence. So what is the mechanism? If we see mechanism, regarding mechanism, basically it's often occur in a specific context such as the mitochondrial RNA in certain organism like trypanosomes. So such type of the organism use this type of the RNA editing technique, or it means where guide in a direct the removal or addition of the uretidine. So uretidine's removal or addition. So that's why it is called indel. So basically this type of the editing is in early phase and not very significant or listener, but the ADRs and CRISPR are very significant editing tool, basically RNA editing tools in these days. So let's see what are the significance of RNA editing. So what is the significance of the RNA editing? So first, if we see, RNA play a crucial role in translating genetic information from DNA into protein. So basically it's translation, as you know, lost in the translation. So if you translate wrong things, then you create such type of the disease. Here is also translation. So translation is one of the most important things in these. So if we see, the RNA editing approach has several advantages. Number one, if we see temporary correction, it is one of the very significant for the RNA editing, benefit for the RNA editing. So unlike DNA editing, which result permanent changes, but RNA allow temporary correction that can adjust if necessary. So you can adjust such genetic disease and genetic disorder. So number one. Number two is less risk of the target effect. Basically, targeting RNA may reduce risk associated with unintended modification. Basically, if you edit DNA, it means some of the base pair unintended changes. So it can cause some difficulty in the patient's or disorder. So that is less risk in the RNA. Another third thing is application in genetic disorder. So successful application have been demonstration in correcting mutation associated with the conditions like Hurler syndromes, cystic fibrosis, Rett syndrome. These are the very lethal diseases like the Hurler syndrome. Hurler syndrome basically accumulations of the large molecules of the sugar. So basically it can damage your lungs, it can damage your hearts, and finally brain. Another is cystic fibrosis. Cystic fibrosis is also affect your lungs, your intestine. So use such type of the cyst and fiber. So these are these type of the genetic condition or genetic disorder, very lethal disorder. So it's a hope for the treatment of the due to the RNA editing. So that's why it is very significant. Another, if you see, what are the challenges associated with the RNA? So regarding these all type of the advantage, some of the challenges. So what is challenges? So if we see, the challenges associated with RNA editing include a specified issue of the ADR. Basically ADR's enzyme, ADAR enzymes may be target unintended side of messenger RNA. Messenger RNA means leading potential side effects. And another thing is its transient nature. It means temporary nature. So basically it's RNA editing required repeat treatment. So if you take some type of treatment, then you come and take this type of treatment repeated one day, after 15 days, after 1 month. So it's repeated treatment. So that is also disadvantage of and challenges basically associated with RNA editing. Another is current delivery system, like the, if you see the nanoparticles and lipids also is a very restricted, very limited. So limitation of the current delivery system. So that's why if you see, in future it will be a game changer in the medical science. So that's why it's very significant application. So regarding, I hope in coming future all these challenges can be addressed by the scientist. So finally conclude it. So I hope I take more time for you. So RNA editing represent a significant advancement of the gene therapy. So if you see, it is a significant advancement of the gene therapy in, if you calculate effect, precision and safety advantage over the traditional methods like the Cas13, Cas9, like the DNA editing and all that, making it exciting area of the future research and clinical. So in future it will be a game changer. So let's see why this is important for the civil services examination. So regarding UPSC, so UPSC question related to RNA editing. So first 3 questions you can easily answer by this video. Discuss the potential of the RNA editing in treating genetic disorders. How does it differ from DNA editing in terms of the safety and efficacy? So number 1 question. Number 2 question, What are the challenges associated with RNA editing? Just we discuss it, all three questions. What is the recent advantage have made in clinical applications of RNA editing, like the clinical applications in the USA, recently approved by the US Food and Drug Administration? So these three things is very significant question for 2025 mains point of view, paper 3rd UPSC. So that's why this video is very significant. Another is regarding the UPSC PT. So please join our test series. Test series is only a nominal fee if you want to join, only 500. And it's a 500, it means 5 set of test series related to all map classes, geography classes and environmental classes. And also you go to our channel and go to the community and see question of the day like this daily question. You can find daily question in our YouTube channel. So thanks a lot. We will meet in the next video in the next time.